the secretion of some factors. Yes, some factors. Which factors? We have three types of factors are releasing here. Like here, inhibin hormones, right? Anti-Mullerian hormones, because it is a testis, right? Anti-Mullerian hormone, inhibin hormones, and ABP. I write here, then you understand. What some factors are there? This is first one is our anti-Mullerian hormone, right? Second substance is our inhibin hormone. Substances also we can say substances. And third one is ABP. What is the full form of ABP? Androgen binding protein. Androgen binding proteins, right? They secretes. What is the function of these three substances which is secreted by the which one? Sertoli cell. Anti-Mullerian means it is responsible for inhibit the that is growth of Mullerian duct, right? Because here all the reproductive ducts in case of male, they are developed from Wolffian duct, not from Mullerian duct, right? In females, Mullerian duct develop into the oviducts or uterus. That is, we can say reproductive duct of in female, they are developed from Mullerian. So this anti-Mullerian means they inhibit the Mullerian duct. They are responsible for the degeneration of Mullerian ducts in male. Second, it is inhibitor hormones. So they gives negative feedback in hormones, right? ABP means what? Androgen binding proteins, that these are responsible for means increase the concentration of hormones. Which hormones? That is FSH in that is seminiferous tubules, right? To increase the spermatogenesis. Now let us examine the structure of a sperm. A structure is given here. The sperm structure is given. This is our NCERT. You can see here, right? So let us examine a structure of sperm. It is a microscopic structure composed of head. This region is head, already given, right? A middle piece. This region is called middle piece, right? This one is middle piece, and a long tail. So three regions are there: head, middle piece, and long tail. In between head and middle piece, this part is called neck, right? So sometimes it is given head, neck, middle piece, and tail, and sometimes it is given only three regions are there: head, middle piece, and tail, right? In head, first stage of head. This head, because it is an animal cell, so it is enclosed in the plasma membrane, which is also given in our NCERT. That is plasma membrane, right? Here, the top part of the head region, a cap-like structure is present, and this cap-like structure is called acrosome. This is acrosome. We can say how, because this is a sperm, so we already know it is given here. A plasma membrane envelops the whole body of the sperm. Sperm head contains elongated haploid nucleus. Haploid means half number of chromosomes, means n number of chromosomes. We can say in human it is 23 chromosomes are present in the head region, right? Anterior portion of which is covered by a cap-like structure, and this cap-like structure is called acrosome. Okay, acrosome. Sometimes it is also called as glia captis, right? This acrosome, it is also called as GALEA, glia capitis, right? This acrosome, it is sac-like structure in which is filled with enzymes that help in fertilization of the ovum. That is called, this enzyme is called sperm lysine. L-Y-S-I-N. Sperm lysine. It digests the zona pellucida, right? The middle piece. Now it is given middle piece directly, right? Here, this is the middle piece. In between middle piece and this head region, there is a neck, and in neck a centriole is present. Look here, this centriole, and this centriole is called proximal. It is not given in NCERT, but proximal centriole, right? Just below the proximal centriole, another centriole is also present. That centriole is called distal centriole. Distal centriole, right? From the base of this distal centriole, right, microtubules are present. They are simply 9 doublets like a flagella, right? Tubules are originate, and these tubules, they are transverse from this whole middle piece and goes into the tail region, right? When they pass the middle piece, there also a ring-like structure is present, and this ring-like structure is called annulus. Annulus means ring, right? And you know already why annulus means what? Ring. Or also it is called as ring centriole, right? Now this filament, it is also surrounded by a spiral, right? We can say spiral is spiral. What is there? Mitochondria. This mitochondria, and this mitochondria sheath is called mitochondria sheath. It is called Nembcum sheath. Nembcum sheath, right? Here, very small sheath of cytoplasm is also present, right? Very less. A peripheral in the form of a sheath cytoplasm is present, and this peripheral cytoplasm means a sheath of cytoplasm, is called here Menschete. Menchete, Menschete. Okay. The middle piece possesses numerous mitochondria which produce energy for the movement of the tail that facilitate sperm motility, which is essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility, what is given here? Normal fertility, at least 60%. At least 60% sperms have normal shape and size, and in this, and at least 40% of them is in 60%. 40% of them must show the vigorous motility, right? Do you understand this one? Now here, suppose that I write here 100 sperms. So what do you mean by 60% sperms? 60% means 60 sperms, right? They have normal shape and size. And what is the meaning of here? 40%. It is not the 40% of this 100 one, but it is the 40% of 60 sperms. What is the 40% of 60 sperms? It is 24 sperms. They must have the vigorous motility, right? Sperm release from the semiferous ducts. The sperm release from the semiferous tubules and are transported by the accessory ducts, different accessory ducts. Secretion of epididymis, first thing, where they mature. Vas deferens, second is given here. Seminal vesicles, which forms the 70% of semen, and in this I already tell you in the previous lecture that it secretes also fructose. Fructose means it acts as fuel for sperms, right? And prostate gland are essential for maturation and motility of sperms. What is even? Secretion of epididymis, vas deferens, seminal vesicle, and prostate, right? Maturation occurs in epididymis, then they transport through vas deferens and then meet with the secretion of seminal vesicle and prostate gland. Already I tell you, seminal vesicle means it is paired gland, Prostate means ring-like single gland, right? Unpaired gland, right? So these secretions are essential for the maturation and motility of sperms. The seminal plasma along with the sperms constitutes the semen. The function of male sex accessory ducts and glands are maintained by the testicular hormones, and these hormones are commonly called as androgens. Okay, androgens. Who maintain the function of secondary male sex accessory ducts and glands? It is testicular hormones. Okay, I already tell you seminal vesicles means it forms 70%. In the last lecture, we discussed about this one. 70% semen is secreted by seminal vesicle, and 30% it is secreted by prostate gland, right? Prostate gland, it is a 5-lobe gland. Okay. Now here, the process of formation of mature female gamete. Process of formation of mature female. Now male gamete means it is spermatogenesis, come clear, right? In spermatogenesis, we read some terms, that is spermatogenesis means what? Formation of sperm. Spermiogenesis means what? Transform of spermatids into spermatozoa. This is called spermiogenesis. Spermiation means what? Release of spermatozoa from seminiferous tubules. That is called spermiation, right? The process of formation of mature, now female gamete, that is oogenesis. Second is our oogenesis. Female gamete is called oogenesis, which is markedly different from spermatogenesis. Totally different. Why different is it? Because in spermatogenesis, it occurs after the puberty, but it is in the embryonic. It starts in embryonic stage, right? What is given here? Oogenesis is initiated. When does it initiate? It initiates at the embryonic stage. It initiates during the embryonic development, or we can say here IUL, intrauterine life. When a couple of million gametes, mother cells, that is called oogonia, right, are formed within each fetal ovary. They are formed within each fetal ovary. How much it is given? Couple of million means what? 2 lakhs in each ovary. That means we can say here they are in total in both 2 ovaries, it is 4 lakhs. Right, 4 lakhs. No more oogonia are formed and added after birth. Right? So they formed before birth. When does it birth? At 25th week. In 25th week, they are formed. Right? These cells start division and enter into prophase first. This oogonia, first what happens? All the cells, either it is a spermatogonia or oogonia, first they develop from a extra, means the gonadial cells, and this extra gonadial cell is called primordial cells, right? From primordial cells, if sperm means in males, what happens? Spermatogonia are formed. In females, oogonia are formed, right? Now oogonia who stores food and become large, and they come into the for meiosis, they are called as oocyte, right? They are called as primary oocytes. There is one primary oocyte. The cell start division and enter into the prophase first of the meiotic division and get temporarily arrested at that stage, and this is called primary oocyte. Means this oogonia. Now it develops and forms primary oocyte. This primary oocyte, it goes into the first meiotic division. First meiotic division, what happens? It is arrested in which phase? Prophase first. Prophase first. In prophase first, we know prophase first is also divided into 5 steps, right? 5 steps means we can say leptotene, zygote, pachytene, diplotin dikinesis, right? It is arrested in diplotin stage, right? Then at the time of puberty, what happens? Then follicle cells are formed. Follicles are formed, right? So when follicles is formed, when the graffian follicle is formed in tertiary follicle, what happens? This first meiotic division is completed in tertiary or graffian follicle. This first division is completed and they forms two cells, right? One is polar body. These are unequal division, right? One forms polar body, another forms, it is called secondary oocyte, right? Now this secondary oocyte, once more, it is arrested in metaphase second. Metaphase second, it is arrested. Then what happens? If fertilization occurs, then the division continues. If fertilization occurs, division continues, right? If fertilization not occur, means fertilization not occur, no fertilization is there, then what happens? It degenerates. But if fertilization occurs, if fusion occurs, then what happens? This second meiosis completes and one polar body and one egg cell is formed, right? Which is fused with sperm. That is here. What is given? Each primary oocyte then gets surrounded by a layer of granulosa cells. I also tell you after this reading, right, how a graffian follicle is developed, right? And it is called as primary follicle. Primary follicle means granulosa cell is present, but antrum is absent, right? Then large number of these follicles degenerate during the phase from birth to puberty. Large number of these primary follicles, they are degenerates. Means at the time of puberty, they are not 2 legs per testis, but remains, they have 60 to 80,000 remaining here. All the other, they are destroyed or degenerates, right? And this is called follicular atreus, right? Atreasis. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovum. How many? 60 to 80. That means in both 120 to 160 follicles, primary follicles are present. The primary follicle gets surrounded by more layers of granulosa cells, means many layers, granulosa cells, and a new theca. Theca means covering is formed. That is called internal theca and external theca, right? We can say theca externa or external. External is made up of, that is muscles. We can say white fibrous connective tissue, and which is made up of collagen fibers. And theca interna, it is made up of cells, granulosa cells, right? It is made up of, we can say this is our cell stromal cells, right? Because it is cellular, what happens? It secretes estrogen hormone, new theca, and these are called secondary follicle. So what is the difference between primary and secondary? In structural difference, in secondary only granulosa layer is there, but in secondary theca externa, theca interna and granulosa, 3 things are there. Means theca is also present in secondary. And when it becomes a tertiary, what happens? An antrum is sac, like a empty space which is filled with that is, we can say follicular liquor, right? It is filled with follicular liquor is present. Antrum present means tertiary. Theca externa, theca interna and membrana granulosa present, but antrum is absent means secondary. If theca is absent, only granulosa is present, no antrum is there, this is called primary follicle, right? The secondary follicle soon transforms into tertiary follicle, which is characterized by a fluid-filled cavity called antrum. This fluid is called, because it is present in follicle, so it is called follicular fluid, right? Or liquid. Theca layer is organized into an inner theca, that is called theca interna, and an outer theca, that is called theca externa. Theca interna, it is a cellular layer, but theca externa, it is a muscular layer which is made up of collagen fibers, or we can say white fibrous connective tissue. It is important to draw your attention that it is at this stage. What happens? That is the primary oocyte within the tertiary follicle. It grows in size and complete its first meiotic division. I write here, right? In graffian follicle, right? What happens here? This division is complete. Graffian follicle means in mature follicle, or we can say these are mature tertiary follicle. Within the tertiary follicle, grow in size and complete its first meiotic division. It is an unequal division resulting in the formation of a large haploid secondary oocyte. Large haploid secondary oocyte means large, what large? Means it has more cytoplasm, right? And a teeny first polar body, and a teeny means very small, in which cytoplasm is very less. Due to the very less cytoplasm, sometimes what happens, it degenerates without division. Sometimes it divide, right, into two another polar bodies, but mostly it is not divide, it is degenerates, right? It is not able. But one thing is equal. The number of chromosomes, it also contains n number of chromosomes, haploid number of chromosomes, and this first polar body, it also contains haploid number of chromosomes. Means number of chromosomes are equal, but quantity of cytoplasm is totally different, right? Secondary oocyte, it has enormous cytoplasm, and this polar body, it has a very teeny protoplasm or cytoplasm. A structure is given here. Secondary oocyte retain bulk of the nutrient rich secondary oocyte because it has more cytoplasm. Due to the unequal division, it retains bulk of the nutrient rich cytoplasm of the primary oocyte, right? Can you think of any advantage for this? What is the advantage for this? Because we know sperms, they have no cytoplasm, right? Very least cytoplasmic sheath is present, which is called Mann sheath, right? So for cell division, the cell needs cytoplasm, right? And this cytoplasm is involved in the further division in cleavage, right? Because we know in cleavage what happens, the cytoplasm, quantity of cytoplasm is not increased, but the number of cell is increased. It needs more enormous amount of cytoplasm, right? This is stored in that. Does the first polar body born out of first meiotic division divide further or degenerate? At present, we have no any idea, right? If it has cytoplasm, means it can divide, but because it have a very least amount of cytoplasm, that due to the least amount of cytoplasm, or we can say due to the insufficient cytoplasm, it cannot divide and it degenerates. At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle, or it is called as graffian follicle, right? So what further change is there? Simply we can say this antrum increase in size, and now this is theca externa, theca interna, and membrana granulosa, they become very thin, right? Also they secrete some hormones, right? The secondary oocyte from a new membrane called zona pellucida. The secondary oocyte, it forms a new membrane which is called zona pellucida, and this zona pellucida, it is a glycoprotein. This one is a glycoprotein which surrounding it. The graffian follicle now ruptures to release the secondary oocyte ovum, right, from the ovary by the process, and this process is called ovulation. So due to the maximum LH, first is there. Second, this tertiary follicle, it also secretes some, that is we can say collagenase enzymes, which are responsible for the thinning and rupturing of ovary, right, at that place from where ovum is released. Can you identify major difference between spermatogenesis and oogenesis? Yes, we can say, because in spermatogenesis, 4 equal sperms, we can say daughter cells are formed, but in case of oogenesis, only one gamete is formed. Spermatogenesis, 4 gametes are formed. Oogenesis, only one gamete is formed. 3 are polar bodies which are degenerate. First thing. Second thing, spermatogenesis initiate after puberty, but in case of oogenesis, it starts in intrauterine life, or we can say in embryonal stage. It has 2 arresting period, means metaphase first, in case of prophase first, right? Prophase first, in meiosis first, prophase first in diprotein, then after another arrest stage also there in metaphase second. But in case of spermatogenesis, this is not occurs, right? A diagrammatic representation of spermatogenesis and oogenesis is given below this figure. Okay, before going to this figure, first here is structure of ovary is given. We see this structure and then we are going to difference between spermatogenesis and oogenesis. What is given here? Look, the outermost. It is made up of simple cuboidal germinal epithelium. Right? Just below this one, it is made up of, just below, this is outer, then this is the below. Just below this one, it is made up of collagen, white fibrous connective tissue, and inside this one, this side, you can see here, right, a very thin portion is there. So this outer is called, it is called cortex, we can say cortex, and this middle portion is called medulla of ovary. In this ovary, this is the blood vessels. So because it is red in color, we can say this one is artery, and this is given in blue color. This is our vein, right? Now here, these are primary follicles. These are primordial follicles. This is the primary follicles. We can see here because it is granulosa layer, right? In primary follicle, what is there? This is granulosa layer, and this is our oocyte, primary oocyte. Now here, because here it is antrum, we can see here antrum. Antrum is there. Because antrum is present, so it is called as tertiary follicle. Antrum is not present here, but theca externa, theca interna is present, then we can say it is the secondary follicle. Here, theca interna, theca externa both are absent, right? But granulosa layer is there, it is called as primary follicle, right? Now here it is graffian follicle or mature follicle, right? Antrum, if liquid is filled up, and this liquid is called follicular, right? Liquid. Now ovulation take place. Look here, ruptured of ovary and ruptured of, we can say, tertiary follicle. Now ovum is released. Ovum contains some cells. These cells are called corona radiata, and a glycoprotein is also there, that is called zona pellucida, and this contains oocyte. Which oocyte? This one is our, which oocyte? It is the secondary oocyte, right? Because here the division is not completed. Here it is arrested in metaphase first, metaphase second. Okay. Now here, this is corpus luteum. First time what happens when it is released? Here blood is present. Here blood is there because it also contains. It becomes visceral, right? It also contains some blood vessels, so it is filled up with blood. So it is called as corpus hemorrhagium. RRHAGIA, corpus hemorrhagium. After then what happens? These cells, they become yellow in color because they produce lutein. Which protein? LUTEIN. Lutein protein, because they contain lutein protein and they are yellow in colors also. They are called as corpus luteum. Corpus luteum Carpus luteum, maximum development of carpus luteum is 22nd day, right? And then after what happens, it start degenerates, and when it degenerates, it convert into carpus albicans. Carpus albicans. You understand this one? Carpus albicans. So this is the structure of an ovary. Where does this carpus albicans goes? Because some cells are also there, phagocytic cells, right? They eat and digest this carpus albicans. It never goes out of the ovary. Okay. Here a means diagram is given for gametogenesis. This side it is spermatogenesis, and another side it is oogenesis. What is given here? Look here, spermatogonia. This spermatogonia come from primordial cell. Primordial cell, they are extra gonadial cells. Means they are not included in gonads. That's why here it is not given. So primordial cells, they form spermatogonia. This spermatogonia, after storing some food, after growth, right? They forms. Look here, what is given? Spermatogonia through mitosis and differentiation. Mitosis means when mitosis, it divides through mitosis and forms two cells. One is called spermatogonia A, and another one is called spermatogonia B. This is A, this one is A, and another one is called spermatogonia B. Spermatogonia B, because two cells are formed, I already tell. Okay, then after this one I tell you, right? Then you understand. This B, through differentiation, it forms primary spermatocyte. This B, it increases cytoplasm and it also increases in size. Then it is called primary spermatocyte. Primary spermatocyte, look here, this one is 2n, because mitosis, through mitosis they are formed, it is also 2n. Here it is also formed through mitosis, it is also 2n, diploid in nature, right? First meiotic division. Now first meiotic division occurs. It has no arresting stage, right? It directly continuously stays there. It forms secondary spermatocyte. How many secondary spermatocytes are formed? 2, 1 and 2, right? Because first meiotic division occurs, and this division is also called as reductional division. So how many? What is the haploidy? Or what is the ploidy of this one? It is only n, right? It has haploid. Then after what happens? Continuously second division occurs. After second division, spermatids are formed, which are circular in structures, right? And their ploidy is also n. And they are also n. Only up to primary spermatocyte they are 2n. After primary spermatocyte they are n, right? This is spermatocyte. After maturation, or we can say here differentiation or transformation, or we here which process is there? Spermiogenesis. Through spermiogenesis, they are converted into spermatozoa. Means now they have tails, and you can see here these are spermatids. These are spermatozoa because they have tails. All these are continuous process. But here what is given? Here in fetal life, fetal means in intrauterine life or in embryonic life, right? Oogonia formed from primordial cell, right? Oogonia is formed. Now here through mitosis and differentiation, it produce primary oocytes. Number of cells are formed, right? These are primary oocyte. It is also 2n. Here ploidy is given here. 46 means what? 2n, right? This 2n. Now primary oocyte, it starts first meiotic division. Look here, in fetal life, this division starts in fetal life, right? Fetal life is before birth, right? Okay, but this first division is not completed. It is not completed. It is arrested, right? It is arrested in which phase? It is arrested. It is arrested in prophase first. In prophase first, which is the sub phase it is? It is diplotin. It is arrested in diplotin phase, right? And it is not completed. At the time of puberty, look here, puberty, what happens? Completed prior to ovulation. At the time of puberty, it starts and it complete before ovulation. Before ovulation means what? When does it complete? This is completed in tertiary, we can say in tertiary follicle, right? In tertiary follicle, this division is completed, and when this division is completed, what happens? A secondary oocyte is formed and the first polar body is formed. This first polar body, either it divide or it will be degenerate because it has a very least amount of cytoplasm. Now secondary oocyte, it starts secondary mitotic division, secondary meiotic division is there, right? In secondary meiotic division, what happens? It once more arrests at metaphase second. These two arrested states, they are absent in spermatogenesis, right? That is a difference. Now here what happens? If fertilization occurs, then this secondary oocyte complete the meiotic second or meiosis second, right? And if fertilization does not occur, then what happens? This secondary oocyte, it degenerates without complete the second meiosis. It degenerates, right? And if fertilization occur, then what happens? At the time of syngamy, when sperm means plasma membrane of sperm and plasma membrane of oocyte, right? They fuse with each other. At that time, what happens? This second meiosis completes, right? And when second meiosis complete means what? The second polar body is formed, which further degenerates, and ovum is formed, or we can say egg cell is formed, and which is fused with sperm, right? That is the difference between these two. What is the difference? First, oogonium is here. That means the formation of gamete starts in intrauterine life in case of oogenesis, and it starts at puberty in case of spermatogenesis. It is a continuous process without any, but it has two arrested phase. First meiosis first, prophase first in diplotin, second in meiosis second, metaphase phase, right? Here 4 equal haploid gametes are formed. Here only one gamete is formed, and here all the division is unequal, but here all the division is equal, right? One thing is equal here, number of chromosomes are equal, right? Now second is given here. Next point is our menstrual cycle. Before going to menstrual cycle, I want to tell you about the development of follicles, right? Then it is to understand the menstrual cycle, it becomes easy. So first come to the development of a graffian follicle. How a graffian follicle is formed? What happens? Here some phase are present, right? Here stages of the formation of graffian follicle. Formation of graffian follicle. What are the stages? The first stage is here. We can say here primordial follicle. It is primordial follicle. In primordial follicle means what? Here it is a primary oocyte, right? Here primary oocyte. That primary oocyte is surrounded by some stroma cells, right? It is that means what happens here? First, this one is primary oocyte, and it is surrounded by some stromal cells. These stromal cells are flat cells, right? These are stromal cells, which are flat cells. And this is primary oocyte. So first what happens? This primary oocyte is surrounded by some flat stromal cells, because these cells, they are responsible for the formation of follicle, and these cells, they are responsible for the formation of follicle. So these cells, this stage is called, these cells are also called as primordial cells, and this stage is called primordial follicle or pre-follicular stage. This one is called primordial follicule or pre-follicular stage, right? What happens then after some time, these flat cells, they become glandular, means they become glandular. For glandular, they become columnar cells. They increase in size, absorb food, and they form a glandular cell. These flat cells, they form cells. Now we can say here, not glandular is not good here, but they becomes columnar cells, right? Then what happens? They start division. One more thing is there. Here zona, because they are columnar cells and they form say epithelium tissue. So here they secretes glycoprotein, and this glycoprotein, it is the formation of zona pellucida. They secretes glycoprotein. And this glycoprotein, it is the starting of formation of zona pellucida, right? Then what happens? This oocyte, right? Here it is the another layer, that is zonal, and these columnar cells, right? They divide and forms a number of cells. Number of layers are cells. 1, 2, 3, 4. Large number of cells. This is called granulosa layer. It is a multi-layered, right? This is our zona pellucida, and this is our primary oocyte, right? Because it contains multi-layered granulosa. Now it is called as primary follicle. This is the second. This is the first stage, and now it is the second stage, primary follicle, right? Primordial follicle, then after primary follicle. Primary follicle means multi-layered granulosa and oocyte. Now this primary follicle, what happens? It increases in size. When it increases in size, it means suppress the stroma cells which are already present in the stroma, right? When it suppress the stroma cells, so a covering is formed of this cell. A covering is formed, right, and of stroma cells, and this stroma cell covering is called theca interna, right? Look here. Now this what happens when it grow, it suppress this is the oocyte, right? This is membrane. This is called zona pellucida. When it grow, what happens? It press this side. Some cells are present in ovary, and what, right? And these cells which are suppressed, they forms theca interna, and this theca interna is responsible for the secretion of oestrogen hormone, right? These are cellular. This theca interna is cellular, muscular, right? Now once more it increase inside, continuously it increase inside, gradually increase inside. What happens? It also suppress outside. What is there? It is collagen fiber, right? This is our oocyte, zona pellucida, membrana granulosa, right? And here a layer is formed, cellular layer is formed, and this cellular layer is called theca interna, and this cellular layer also suppress the muscular layer, and this muscular layer, right? These are the muscles, muscular layer. This muscular layer, it is called theca externa. This one is theca externa, and theca externa is made up of which muscles? W, F, C, T. White fibrous connective tissue means collagenous, collagen fiber, right? This is cellular theca interna, right? It is cellular, made up of cell. Already I read right here. Cellular, right? This I like. So when theca externa and theca interna are formed, this is granulosa layer, and this one is zona pellucida, and it is primary oocyte, right? So this structure is called secondary follicle, right? Primary follicle, second step is secondary follicle, third step is secondary. Primary means first step is primordial follicle, second is primary follicle, third is secondary follicle. In secondary follicle, what is there? It has theca externa, theca interna. Secondary, primary follicle means this theca externa, interna is absent. Now what happens when it grows inside? What happens? And a cavity is formed in granulosa. A cavity is formed, and this cavity is called antrum, right? Look here. Now it is the zona pellucida. When it grows, a cavity is formed, right? This is membrana granulosa. In membrana granulosa, a cavity is formed, and this cavity is called antrum. Antrum, right? In this cavity, a liquid is present, and this liquid is called follicular liquor, right? And other things are also present here. This one is called what? Called theca interna, and outermost means muscular layer. Muscular layer is called theca externa, is also present here. This is theca externa, right? Now this, because antrum is present here, this one is called tertiary follicle. Tertiary follicle, right? In antrum, it is the characteristics of tertiary follicle. One thing is here, and this is the tertiary follicle, means here meiosis first is also completed. Meiosis first completes in this phase, right? Now the third one, then after what happens here? Look here, this tertiary follicle, it increases in size. It increases size, and what happens here? This antrum is also increases, because antrum is increases, right? What happens? This centrally arranged. It is centrally arranged, right? Here it is this our oocyte, right? It is centrally arranged, but now it becomes eccentric. Means it comes to one side, right? It becomes eccentric. Some granulosa cells are attached with oocyte. Now it is here. Now I call this one secondary oocyte, because I tell you here, this meiosis first is completed here, right? After meiosis first complete, what happens? Secondary oocyte is formed, right? So secondary oocyte here, secondary oocyte, it located eccentrically, right? Some cells are also attached with secondary oocyte. These cells are called corona radiata, right? And the cell which are present at the antrum side, they are called as cumulus or follicles, and some cells which form the disc, or which we can say they are attached with other granulosa cells, they are called as discus proligerus, right? Okay, I draw a structure and you can understand from this structure. Now this is our mature graffian follicle. Outer side it is attached with the fibrous connective tissue, white fibrous connective tissue. This tissue is called white fibrous connective tissue of ovary, right? And this is the also white fibrous connective tissue of follicle, tertiary, or we can say graffian follicle. It comes near to the, right? Okay, what happens here? Then after what? We have theca interna. This is the theca interna. Theca interna. This WFCT of means it is theca externa, right? Then after granulosa layer is there. These are granulosa cells, granulosa, membrana granulosa, right? Look here, this is the oocyte. Now location of oocyte is eccentric, right? Here zona pellucida, right? This side, these cells, these are called as cumulus oophoricus, right? And this is the base side, this is called discus discus proligerus, proligerus, right? These cells, they forms, which is cumulus oophoricus, some cells, right? They forms after the ovulation, they forms corona radiata. This layer, this single layer, this layer is called zona pellucida, which is a glycoprotein. And this is our, now it becomes secondary oocyte. That is very important. It is not a primary, but it is secondary oocyte, right? And this is the antrum. Okay, so these are the different, we can say these are our different stage of a developed follicles. You can see here, look, we can start from this one. Follicle stage of formation, first primordial follicles are present, right? Now they forms, this is our primary follicle. When theca interna and theca externa is developed, now it is called as secondary follicle. When antrum is present, it is called as tertiary follicle. And in tertiary follicle, what happens here? First meiotic division is completed, right? Now it becomes this oocyte, it becomes the secondary oocyte. And at last, what happens? This tertiary follicle is converted into graffian follicle or a mature follicle, right? In mature follicle, this oocyte is not a centric, but it becomes eccentric, and it has some cells toward the antrum. These cells are called as cumulus oophoricus, and some cells toward the granulosa layer, which forms the disc-like structure, and this is called discus proligerus, right? This is our mature graffian follicle. This is the development stage of graffian follicles, right? Okay. Next chapter, I think this our time is over. In next lecture, we will start from menstrual cycle.